All right, hello. Let's talk proteases. Proteases are enzymes that cut protein. And this is another trick of biology. Anytime you see something and it has ASE on the end, that means it's an enzyme. And usually it's an enzyme that cuts whatever is at the front of the word or chemically modifies whatever's at the front of the word. So a protease, yeah, cuts protein. A DNase, what do you think that does? Cuts DNA, right? Yeah, of course, yeah, yeah. So this is what a protease does. And if you notice, it's actually the same way that we build proteins. We're just reversing the process. So you can call it a condensation because we're bringing two things together, or you can call it a dehydration because we're taking out of water to make the protein down here plus water. And then you take the protein and add a water molecule, and so it's a good thing that we're mostly made of water. But you add that water molecule in, and you can actually break the protein apart again. That's what a protease does. It pushes this way, whereas other enzymes that are building proteins are pushing in the opposite direction. So, there are a lot of proteases that are very important to you, and the biggest class of these, the most widespread and widely useful in our bodies anyway, are called serine proteases. Now, serine, you've probably seen that word before. That's one of the amino acids, right? It's the amino acid that's called S, yeah, or abbreviated to S. And so it's got a carbon with two hydrogens and then a hydroxyl group at the end of it. So it's going to be one of these polar amino acids. And that little side chain, that oxygen right at the end of serine, is actually the driving force for the chemical reaction. So there are many forms of these serine proteases, and a serine protease is actually a dangerous thing because your cells are mostly made of protein, and proteins are all the little machines that keep your cells alive. And so, if you have a loose protease that's going around and cutting all the little machines that are the cell skeleton and do all the enzymatic reactions inside the cell to make and break ATP and just keep everything moving, then you will have a dead cell. And so we secrete these proteases in an inactive form, meaning they don't work in the form that we start off with. And then each of these things is going to get cut, usually by a different protease, and that will then turn into what we call the active form. So, for example, trypsin is one of your. Actually, trypsin and chymotrypsin are digesting your food right now. These are enzymes that are secreted to break down protein in the food that we've eaten. But we don't just make them in that form because that would be dangerous. We kind of, it's kind of like throwing a grenade. You pull the pin just as you let go of it. Oh, well, not with a grenade, but with these things. So just as they are outside the cell, we then activate them and let them act on the food rather than tearing up the cell they just came from. Elastase, though, is a neat one. This is going to be involved in blood clotting. And the other thing on here is that, so they're all secreted from the pancreas. That's great. Oh, actually, no, these are all, I'm thinking of prothrombin and thrombin. Sorry. These are all digestive enzymes. And the neat thing is that they each go after different amino acids. So trypsin will go after and cut right after it finds one of its favorite amino acids, which is going to be an R or a K. Now, single letter amino acid code knowers that you are, what's an R? What's a K? And what do these two amino acids have in common? I'll give you a sec. Or pause it. So R and K, R is arginine, K is lysine. They're both basic. They both have a positive charge on them. Trypsin is looking for an amino acid with a long side chain and a positive charge at the end of that. And that's exactly right. That's exactly what it's trying to bind. Chymotrypsin is going after W, Y, and F, and you can say, okay, what do these guys have in common? W, Y, and F all have rings in them, And these are all aromatic rings. So these all have electrons that can move from one place to another and exist in different forms. These are very large, very hydrophobic amino acids in general, although the tyrosine does have, that's the Y, does have a hydroxyl at the end. And so it does sometimes get included on lists as being polar. In my class, you can call tyrosine either polar or hydrophobic, and you're mostly correct either way, I would say. It's one of those that's kind of right on the line. And then elastase over here is looking at G, A, and V, and so if you looked those up, you would find those are all hydrophobic amino acids. And if you looked at their picture, those are the three smallest hydrophobic side chain amino acids. So that's what elastase is going after. And the original form that's inactive is called a zymogen. That's the like the trypsin-ogen form. So ogen is a zymogen. That's just kind of the ending we put on it so that we can tell what's what. And just like in reality, trypsinogen has an extra little piece of protein that you have to cut off to make it into trypsin. And the word trypsinogen has a little extra part on the end, the ogen, that you have to cut off to make it the active form, trypsin. Oh my gosh, yeah. It's names reflecting reality, for once. Okay, so how do different proteases recognize their substrates? The answer is each protease has the same substrate specificity pocket, but they'll have different amino acids in that pocket that will interact differently with the side chain of the thing that they want to cut. And so for chymotrypsin, which is going to go after these big rings that have distributed electrons, they're aromatic rings, it's going to have a very wide open pocket with little hydrophobic things on either side to sort of interact with that electron cloud. And for trypsin, which is going after the positive things, it's putting an aspartic acid, which has a negative charge down at the bottom. And so negative and positive opposites will attract. And for elastase, it's looking for something very small and hydrophobic. And so it's actually going to block the pocket and put a valine at the top, which is hydrophobic. And it's going to interact with a small thing like an alanine, which is shown here. Okay. So, yeah, trypsin can actually, so chymotrypsinogen is the thing that's going to become chymotrypsin eventually. And the way that this gets activated is that trypsin is going to cleave one peptide bond. So here's the whole protein from the start to the end, 1 to 245. It's going to get cut by trypsin, and now it's going to be called pi chymotrypsin. It's going to be active. And then it's going to be cut by pi chymotrypsin. So one is going to cut another pi chymotrypsin molecule. And you're going to end up with two little chains. And this is going to be called alpha chymotrypsin. I believe it's the part in the middle that has the enzymatic activity. And yeah, that is also a nice, very active version. So there's different levels of activity, and so this is how we can control the different level by just how far we allow the cleavage to go. Blood clotting also involves serine proteases. Pretty much everything in the cell involves a serine protease in some way or another, yeah. But very definitely in blood clotting. So thrombin is the name of one of these serine proteases. And it's going to take a chain called fibrinogen, and it's going to cut it to form fibrin. And fibrin actually makes the little filaments that make your blood clot. So this is, these are like a little net that then catches red blood cells and stops them from just leaking out of your body indefinitely. And so thrombin is going to be, very specific, and it's only going to cut the arginine to glycine bonds in a specific sequence and structure context. So the thing that is going to be cut has to be somewhere that's exposed, somewhere accessible to the protease. Because the place in the protease where a protein gets cut is actually kind of down the inside. It's kind of like imagine your hand in a big winter glove and now fold it up, like make a fist out of it. The active site is right down in that little crevice, like that little valley in between your fingers and your palm, basically. And so whatever it is that's going to be cut has to be able to fit into that part. And so you can have the same sequences, but they're out and they're part of a surface, like a big broad surface like a soccer ball, and that just does not fit down into the active site, and so they just do not get cut. All right, practical use of these things. Why not? Snakes use them, yeah, all the time. So, snake venom from the most venomous vipers, like this little guy here, will contain 5 to 10 different serine proteases in a single venom. One of the things that they do is to either cause random blood clotting, just make all the blood clot, like activate that prothrombin, or they can cause blood to not clot. They can cut something like the fibrin, so that even if you try to make a blood clot, you can't, and you just start to bleed out. Yeah. These are both ways that the snake is trying to basically get you under its power so that it can eat you. Now, it's by you, I mean usually a little mouse, but the venom works the same in people. It's just that there's no payoff for the snake at the end because they cannot fit you into their little tiny mouths, even if they dislocate their jaw all the way. All right, enzymes work because they have a little tiny region, usually at the bottom of a pocket, like a little cleft in the protein, and this is going to be called the active site. And there will be usually 2 or 3 amino acids at the bottom of that active site that are actually going to do the chemistry. And the chemistry is going to happen because of the side chains of the amino acids, so not the backbone of our protein. Just the different side chains will give it its enzymatic powers. So, we're going to go through the mechanism here, and then I'll put a video out where we go through a YouTube video that shows the mechanism again, just the same as here, but with a little movement. And your goal from this lecture is going to be able to take the starting point and work forward, or take the midpoint and work forward, and tell me what happens next, all the way until we get to the end of that section. So you'll only have half of the reaction mechanism, but you won't know which half, and some of you will have the front, some of you will have the back, so you need to know the whole thing. It just makes it a little easier to grade. All right, so important terms. Catalytic triad. The triad means 3 things, so these are the 3 amino acids responsible for catalysis, which is making the chemical reaction possible. And the chemical reaction here is, since this is a protease, the reaction of breaking a protein at one of those peptide bonds. The oxyanion hole. So break that word down. Oxy comes from oxygen, and anion is a term that you might remember from chemistry or just living in the world. This means an ion with a negative charge. Think of it as like a negative ion. Anion, yeah. Positive one is a cation, and don't worry about cations. We don't have any of those in this particular diagram. Yeah, so oxygen with a negative charge is an oxyanion, and the oxyanion hole is a place where this oxyanion can go for just a second. Because oxygen with an extra electron is going to try to react with the next thing that it sees. It is a very chemically active group at the normal sort of conditions that you would find inside of a cell. Last bit we've already actually covered is the specificity pocket. This is the little hole that binds the substrate and repels other molecules. So it's going to lock in the amino acid that's going to get cut. Almost as if you went in for a haircut and they sort of clamped you into the chair before cutting your hair. That'd be weird, but that's kind of how a protease works. So the active site for chymotrypsin is going to be these 3 amino acids. And you don't have to remember the numbers, but you are welcome to remember the numbers because they are correct. Yeah. So the serine 195. This is going to be the active molecule, and this is the serine that gives it the name serine protease. There are other serines in a chymotrypsin molecule, but they do not participate directly in cutting a protein, and so we pretend they don't matter. This is one. There are serine proteases. There are other kinds of proteases, like metalloproteases would use a metal ion to do the cutting, for example. But yeah, this one's a serine protease. And so you can see right now it starts out with the serine, which has that CH2 group here. And so imagine the protein backbone is right here where I'm moving my arrow. And here's the hydroxyl hanging off at the end. And you can see that the hydrogen, which is actually bonded to the oxygen, is also hydrogen bonded to the nitrogen. So the nitrogen is looking at it and thinking, maybe I'll steal this one. Yeah. And so this is our histidine in the middle, and it's histidine 57, which I think of as like Heinz 57. I don't know. Whatever, it works. Yeah, it's a ketchup. Yeah. And then over here, the histidine already has one hydrogen of its own. But even so, it's looking to steal this one because there's an aspartic acid over here, which has its eye on this hydrogen. And so it's going to try to steal this one. And each of these little red lines is a hydrogen bond. So there's an interaction, but it's not a covalent bond. It's just an interaction where two things will sit. They'll have a energy minimum at a particular distance from each other. Where the basically attractive and repulsive forces are in balance. And so that's where they stay. And they don't want to move out of that spot. Okay, structural similarity. So we mentioned three enzymes, trypsin, elastase, and chymotrypsin. These are the backbones, so we're pretending there's no side chain on here, of all three enzymes. Do you see the resemblance? So trypsin's yellow, elastase is green, chymotrypsin is, it almost looks gray to me, but they're saying blue, so fine. And yeah, look how they just run right on top of each other. There are a lot of places where you can only see 2 out of the 3 because 1 out of the 3 is actually under the other 2. So these are very similar proteins. We give them different names as if they're totally different, but quite similar. The big difference is in the specificity pocket, which I think might be right over here. I think that might be the pocket. Either that or it's right here. No, it's not right there. I think it is right over here. Yeah. And you can see those 3 active site amino acids. So this is the serine, this is the histidine, and this is the aspartic acid right over here. All 3 of these enzymes have the same the same catalytic triad, and they are actually positioned very, very similarly in all 3 molecules. And that's the only way that this works. Everything has to be very close together and at just this angle and in just this way in order for you to have a working protease. So here's the mechanism. We'll do the less entertaining version here, and we'll save the more entertaining version for the other video. And what you want to do with this, I would say, is come up with a story or come up with a logic that you can follow through to the end. So in this, I think of oxygen as basically always wanting more. Always trying to steal an electron, and when it has an electron, always trying to use that to basically harpoon another atom. And just, it's basically, I don't know, kind of like that. It's very reactive. It would like to bond with something, and it would prefer not to bond with hydrogen because hydrogen does not have a lot to offer, let's say. And so here's our hydrogen bonds shown in yellow. Here's our histidine, and here's our aspartic acid right over here. Looking great. So, step 1, a peptide is going to come in and bind to the active site. And that's exactly what happens. Okay, step 2, and I might need a little more explanation than we can see up here, but take notes from this and add a little bit onto this if you can. So the first thing that happens is that the R1 group, so this is the rest of the protein, but it's the front half of the protein. So we're going to cut after the front half of the protein. This is going to bind right down here, and this little lump is the specificity pocket. So this is where we check out and see if this is the right peptide. And so the specificity pocket will accept a particular amino acid and will repel other amino acids from this. And so this is how we control what the protease is able to cut. And also what the specificity pocket does is it basically holds the protein in place while the chemical reaction takes place. So it's going to lock this little protein into position. So R1 is the beginning of the protein, R2 is the end of the protein, and this carbon to nitrogen bond is the one that we are going to cut with the protease. So the first thing that happens is a series of attacks. And by attacks, I mean this in the chemical sense. This is atoms forming bonds. So this oxygen has a negative charge. It's actually going to attack and steal this hydrogen. We've left off the little arrow here for some reason, but that's going to happen. This nitrogen has an extra pair of electrons, and so it can take this hydrogen on. That's fine. And that would give oxygen back its electron. And so nitrogen takes the hydrogen. Oxygen has an extra electron, which makes it an oxyanion, O with a negative charge, because there was one electron involved here in this bond. Oxygen will then use this electron, and it will basically stick it into the closest thing, which is going to be this carbon right down here. And so oxygen, by attaching its electron to the carbon, is actually going to make another bond. And so our oxygen will have a bond on it. And oxygen likes to make two bonds, so it's going to stay bonded here. It's going to bond to the carbon, but it can't hold on to the nitrogen or the hydrogen, so that's going to go over here to the nitrogen. Now, carbon can only make 4 bonds, and so we just made the 4th bond, and you see this oxygen is double bonded up here. And so one of these 4 bonds has to break, because one of these 5 bonds that we've made, the 5th one is right here. Just have to imagine it's where this pink arrow is showing. That electron is going to join oxygen to carbon. And so what ends up happening is that this oxygen gets back an electron. And so this is the next step. So now we have oxygen bound to carbon. So it used that electron to bind here. We have hydrogen over here. And this oxygen is holding on to this hydrogen right over here. Next step is that the oxyanion hole is going to temporarily stabilize this form, this oxyanion, so that the reaction can proceed. Then after a second of stabilization, the electron is going to come back down here, and we are going to reform that double bond, because oxygen is going to basically just attack the carbon with this electron and make that double bond back. And so now, once again, we're going to have 1, 2, 3, 4, and the 5th bond that we just made here. And so the question is, which one are we going to get rid of? And the answer is that nitrogen would like to have those electrons more than anybody else. So it's going to take its electrons back. It's going to pull the hydrogen off of this histidine, and this nitrogen is going to get its electrons back. And so we're going to end up with hydrogen here, nitrogen with its electrons, and yeah, the rest of the protein, the front half of the protein, stuck to serine 195. Just like so. And now that the other half of the protein is free, the second half of the protein can leave. So this is called the amine product, and it can leave, and it can go somewhere else, get cut by some other protease or have some other function, whatever. Okay. Now that we are here, the next step, and so look, we're kind of at a crossroads. So we've got our serine, and look, serine should have CH2 here, and it should have OH here. But look, we have half a protein chemically joined, covalently bonded to our serine, and that is not good. That's not good at all. Yeah. So we need to do something about this, and yeah, we basically need another molecule to come in and help do something. So let's find one. Okay, so that was the end of the first half. The second half starts when this molecule comes in, which is water, H2O. And so there's water all around, and so this is relatively easy. There's going to be a water molecule, and it's going to be attracted in. The nitrogen up here with its extra electrons is going to try to grab this hydrogen and use it to make a bond with those electrons. And so it'll do that, which gives you OH minus, and the minus is going to be on the oxygen. So once again, we have an oxyanion, and over again in this reaction mechanism, what an oxyanion does is it will take that electron and it will stick it into carbon to try to make a bond. This is oxygen's main goal in organic chemistry, is to bond to a carbon somewhere. Oh my gosh. Unless you absolutely take away that option, then it'll do other stuff. But that's the really common thing that happens. Okay, so oxygen is going to bond to the carbon. Carbon is now going to have 1, 2, 3, 4 that it already had, plus 5 bonds. Carbon can only take 4, and so it's going to reject one of these bonds. This oxygen is going to get its electron back for just a second. It's going to be an oxyanion. It will be stabilized once again in the oxyanion hole. So this is a thing called the tetrahedral transition state, and it's going to exist for a very short amount of time. A typical enzyme will do the entire reaction on the order of a picosecond to a nanosecond. So let's see, a billionth of a second to a trillionth of a second. So very fast, very, very fast. So the oxyanion will then come right back down and make a second bond here, because what else is it going to do with that electron? There's no good carbons to stab into, and it can't quite reach that one. And so now our carbon has a problem again. It's going to have 1, 2, 3, 4 that it had, plus one more that it just made, and carbon needs 4, not 5. And so carbon is going to push back the basically the weakest link. And this oxygen is going to get its electron back, and it's going to be an oxyanion for just a second, but it's going to use that negative charge, that extra electron, to grab onto the hydrogen. And so that's going to pull the hydrogen away from nitrogen, which was happy keeping its 2 electrons to itself beforehand. It didn't really need the hydrogen, but it can take the hydrogen. So yeah, and we're going to end up right back where we started. We're going to have a serine with an OH, hydrogen bonded to the histidine, hydrogen bonded to the aspartic acid. And now the other half of the protein is free, and so it can leave. It can go right out of the reaction center of the enzyme. So watch this, maybe watch this several times, then watch the other video, and yeah, the idea is to come up with a way to remember these things. And I think there are pretty good ways. I like to picture oxygen and carbon and hydrogen and nitrogen as people. And this is some kind of drama, basically. And I think that works. Or, yeah, you can substitute whatever you want. You can put your own friends into this. Oh my gosh, it's fine. Whatever you want to do. All right, so last little bit to finish up a bit about serine proteases. Yeah, since we know a bit about protein structure, serine proteases actually all share a common structure, and it looks like this. So on this side, we have a thing called a beta barrel. You see all these beta strands here are twisted around each other. And over on this side, we have another beta barrel. Yeah. And these beta barrels are called anti-parallel because this one goes this way, this one goes this way, then this one goes this way, that one goes the other way. And so one goes up, one goes down, one goes up, one goes down, just like that, over and over. And they're wrapping around. We're looking down the barrel. In this case, we're looking at the side of the barrel over here. And there's a little alpha helix hanging up here at the top. Yeah, so there's 2 domains, each of which is a beta barrel domain. And beta barrels are pretty common in the world. The active site is actually in the crevice right between those 2 domains. And so you've got your, let's see, serine, you've got your histidine here, and you've got your aspartic acid. Now you see these each come from different parts of the protein. Histidine was number 57, aspartic acid was number 102, and was serine number 195? Let's check. Yep, serine number 195. So there it is, right there. All right, so if you're thinking in evolutionary terms, like how could we end up at the biology that we can see in front of us? We've got similar amino acid sequences between all serine proteases. So they're all probably just duplicates of one another that have duplicated and then changed a little bit at some point of that process. So duplicated and diverged. Yeah. So yeah, since they have very similar structures and similar sequences, they probably came about from divergent evolution, where you start with one thing, and then the children look a little different than their parents until you get 2 things that are different enough you can notice, basically. And we would also say that these probably evolved from gene duplication. So it was probably a single beta barrel protein that was doing who knows what that got duplicated and just happened to have these amino acids on the 2 barrels, and they're now in the perfect position to actually do some chemistry and be a protease. That's the idea. There's another protease, though, that is a bacterial serine protease. It's called subtilisin from Bacillus subtilis. It's a bacterium. And this is something that they will use in what are called biological laundry detergents, or enzymatic laundry detergents, I think would be more common in this country. And yeah, you can get these in the regular store. They have active proteases inside them. And so this is how they get out tough protein stains by physically, going in there and cutting the protein. So yeah, that's kind of neat, right? And so yeah, there's a lot of commercial interest in making proteases that have different properties. So they can work at high pH, because most soaps are rather high pH, or can bind to different things, get out a different stain. The only reason the stain is still there is because some molecule or potentially some protein is left behind on the cloth. And if you can attack whatever that protein or molecule is with enzymes, you can physically just break it down to the point where there will be no stains there. So yeah, for your whites being whiter than new and your colors being brighter than new, there are serine proteases. Isn't that nice? Okay, so how does subtilisin compare with chymotrypsin? Well, they're in a different family, in a different clan. These are ways, kind of old-fashioned ways that we group serine proteases. The S at the front tells us it's a serine protease. But interestingly, even though these are both serine proteases, they have no detectable sequence similarity. And actually, their structures look totally different. Yeah. The structure is... on subtilisin is a thing called an alpha-beta fold or an alpha-beta sandwich, and it looks like this. So you can see here, imagine this is a sandwich. This is like a hamburger that's standing on its side for some reason. The alpha helices at the top and bottom, those are like the bun, and the beta strand running through the center, that little beta sheet, that's like the meat. So the beta sheet's the meat, and the alpha helices are the bread. There we go. And so here it is, looking at it from the top, you can see the alpha helices on top. And you get a little bit of burger poking out, like in any good burger commercial. They'll show a little bit poking out. So compare these two things. Here's our double beta barrel structure with those 3 amino acids, the serine, histidine, and aspartic acid. Here is our alpha-beta sandwich with 3 amino acids. Look, it's the serine, here's the histidine, and here's the aspartic acid. These are both the same kind of serine protease. All the rest of the protein, its job is to form a surface that can attach to other things, and its other purpose is to get these 3 amino acids into exactly the right position to be able to do this reaction. And as you can see here, it doesn't really matter what those amino acids are, what shape they make. Their job is to make a shape that gets these 3 things into just the same order. And so those are superimposable. You can line up those 3 amino acids and they're exactly the same on the 2 enzymes. It's just that everything around them is different. It's like, like an inactive ingredient, if you like, the outside, and the little active ingredient right here. Okay, so yeah, same catalytic triad, different order in the sequence, and different, completely different appearance of these things. And so we would say something like this is probably the result of convergent evolution. So there's no good way for a double beta barrel protein to turn into an alpha beta sandwich protein, because all the steps in between would involve different structures that probably wouldn't keep those 3 amino acids in the right orientation to be able to do the chemistry. So we think this is a different protein, like an alpha beta sandwich protein that, one day happened to make just the right mutations to end up with a serine, a histidine, and an aspartic acid right at the perfect positions to do proteolysis, to be a protease. So, yeah, I don't know. All right, so now that you've seen this, can you tell me what a serine protease is? Like, what is it? What does it do? It's like a protein-cutting enzyme that uses serine at the reaction center to actually start the reaction. I would say that's a good one. Can you give some examples of serine proteases? Oh my gosh, yeah, I can. Yeah. We had, trypsin, chymotrypsin, elastase, thrombin, and we had subtilisin at the end. Key aspects of the structure and function. Yeah, so do you know what that structure is? Which of those enzymes has the double barrel fold? And which of those enzymes has the alpha-beta sandwich fold? Interesting, and please make notes. And then, based on this, what are some evolutionary notes? Like, how do we think this could have evolved? how you could end up with a situation like we've got right now. So the one is probably, or rather, the similarity is probably convergent in one case and divergent in the other cases, I would say. So there we go. That's the end of this lecture. Thanks very much.